introducing all of our amazing speakers, but first there are some housekeeping items I just wanted to briefly cover. So as a reminder, this is a hybrid workshop. We have a number of folks participating via webcast today, so the agenda is available on the workshop webpage. Please also refer to the full biographical summaries of our speakers posted on the workshop webpage to learn more about their amazing work. Following the workshop, meeting materials, including recordings, speaker slides, and also transcripts will be available online, so be looking out for that sometime within the next 3 weeks. We have Wi-Fi availability for all of our in-person guests, and that information should be featured on the last page of the printed agendas, which also includes a QR code, which will link you to some of the meeting materials. So additionally, refer to our housekeeping slides that will be displayed during the breaks. We have food at the kiosk right outside of this room in the main hall where you can purchase coffee, assorted beverages, and also snacks throughout the day. If you would like to order lunch, please do that and submit your order by 10 a.m. at the kiosk. Again, that's located in the main hall. And you can pick up your lunch during the lunch break. So lunch can be eaten within the room or at any tables at the main hallway, or if weather permits, at one of the tables outside. Restrooms are also located in the main hall behind the kiosk. And if for any reason you need to leave the FDA building, just be mindful that you will have to go through security once you re-enter. And lastly, we have already received a number of public questions during the registration period, and many of those questions will likely be covered during the workshop, but if time permits, we will try to address any additional relevant questions to our speakers during the panel discussions at the end of each session. So with all of that said, I am now happy to introduce our first speaker, Dr. Terry Michele. Dr. Michele is the director of the Office of Nonprescription Drugs here at FDA, and she'll be delivering some opening remarks on today's workshop. Thank you.
So good morning, everyone. Now, it is just such a pleasure to welcome everyone here with many of you in the room as we get back to in-person meetings at White Oak, as well as all of the many people attending virtually. Just in time for Halloween, it's my pleasure to have you here at this workshop today on candy-like dosage forms. So there's a natural tension in formulating drugs between creating palatable dosage forms that people, and especially children, are willing to take, and the potential safety and manufacturing risks that occur if you make those forms taste too good. So we're trying to figure out what that means today. And candy-like dosage forms for non-prescription drugs aren't a new phenomenon. They've been around for many years, with even Mary Poppins singing about how a spoonful of sugar helps the medicine go down. However, we've seen a proliferation of these products over the past few years, particularly in the dietary supplement market, and now it's starting to creep into non-prescription drug products as technology provides increasing options for how we can formulate drugs. So given this, we started looking at what information exists on these types of dosage forms, including what information exists on consumer understanding of the dosage forms and the potential confusion that might exist between these dosage forms and candy. We realized pretty quickly that there's just not a lot out there, and with very little research, starting with something as basic as a definition, it's just not there. So since a first step in starting to gather data about this is defining the products that we want to know about. We realized that a we know it when we see it kind of approach just wasn't going to cut it. And so the idea for this workshop was born. The number one goal for the workshop is to further define features of non-prescription drug products that could be considered candy-like. So to help us come up with a definition for what these formulations are. And as such, we'll be exploring this question with each of our panels to get ideas on this from all of the experts. And in addition, we'll be exploring formulation and stability considerations, implications for adherence, the potential risks of these dosage forms, things like accidental overdose, especially in children, misuse and abuse of these types of dosage forms, GI side effects, effects on blood sugar, dental issues raised by the high sugar contents and sugar substitutes in some of these products. We are honored today to have an incredible lineup of speakers with representatives from industry, from academia, from the U.S. Pharmacopeia, from state poison control centers, and from federal agencies, including CDC, as well as many of our colleagues here at FDA. I'd like to thank all of our speakers today for donating your time and all of your expertise to this important effort. I'd also like to thank our collaborators from the Division of Pediatrics and Maternal Health, the Office of Clinical Pharmacology, and the Office of Pharmaceutical Quality here at FDA for helping put on this workshop, in addition to our collaborators from the University of Maryland Center for Excellence in Regulatory Science and Innovation, without whom this workshop could not have taken place. And last but not least, I want to give a big shout out to Brandon McClary, an interdisciplinary scientist reviewer in the Division of Nonprescription Drugs 1, who you heard from already this morning. He is the mastermind behind this workshop. He's put in countless hours of tireless dedication, along with others from our group as well as others, for more than a year to bring this workshop together. And this is on top of all of his other work. So it's a real testament to the dedication to public health to bring this together. So thank you, Brandon. And finally, I'd like to thank everyone in the audience for attending, for adding your voices to this important dialogue. We look forward to the thoughtful questions that we've been already have coming in during our panel discussions. And with that, I will turn it back to Dr. McClary.
Thanks again, Dr. Michele, for your remarks. So we will now begin our first session, which is titled Formulation Considerations for Solid Oral Candy-like Dosage Forms. And first, I'd like to introduce Dr. Natalia Davydova, Principal Scientist with the USP, the United States Pharmacopeia. She will be starting off our first session, giving an overview of USP monographs for chewable gels.
Good morning, everyone. My name is Natalia Davydova, and I'm scientific liaison for dietary supplements, finished dosage form, including chewable gels, which represents gummy products. I am glad to have the opportunity here to present USP activities in the developing of monographs for chewable gels marketed as gummies. How I can make it? It's back. Okay. USP currently developing chewable gel monographs for dietary supplements only. Dietary supplement chewable gel continue to gain popularity in market among a wide range of the population, from children to elderly, due to their pleasant taste, attractive appearance, and ease of intake. Based on the chart, the U.S. gummy market is expected to grow at a compound annual growth rate of 10.8% from 2022 to 2030. Is important, therefore, it is important that dietary supplements manufacturers produce high-quality chewable gels that are safe and effective to deliver the intended nutritional benefits indicated on the product label. In my presentation, I will provide some overview of chewable gel monographs, monograph components, and specifications given in the monograph, which indicate USP quality attributes for this dosage form. Also, I will touch up potential safety issue and recommendations for consideration when developing the chewable gels. We have currently 4 official monographs for chewable gels covered gummy products. This is ascorbic acid, cholecalciferol, cyanocobalamin, oil and water-soluble vitamins with mineral chewable gels. We also ready to publish ubidicarinon chewable gels in the near future. Also, we have more monographs under development. We currently reviewing submitted documents from the manufacturers. Here in some of the example of the monographs, future monographs. So the first component of our monograph is the title. And we were not able, the title of the product of the monograph covered gummies is chewable gels. We were not able to initiate the development of monograph before the definition of this dosage form was defined in the General Chapter 1151, Pharmaceutical Dosage Forms. Since it was, it was quite questionable if this finished dosage form can be a good dose or acceptable dose for delivery, not only dietary ingredient, but also drug compounds. Finally, USP Nomenclature Expert Committee agreed to introduce the name chewable gels for monographs covering dietary supplements, commonly called gummies. General Chapter 1151, Pharmaceutical Dosage Form, has been revised and included information on chewable gels, a new dosage form for oral delivery of dietary supplements and drug substances. Here is a partial definition, I could say, from General Chapter 1151. Chewable gels are used to deliver drug substance or dietary supplements via the oral route. In addition to the drug substance or dietary supplements, chewable gel can consist of all or some of the following components: gelling agent, sugar, water, sweeteners, and flavoring agent. The sweeteners and flavoring are intended to enhance patent acceptance and mask the taste of the delivered labeled drug substances or dietary supplement. Chewable gel maintain their molded shape, are elastic, and yield to mastication. They are intended to be chewed before swallowing. Chewable gels are also known as gummies in the confectional and dietary supplements industry, but that item is not used in official article title. The next, as soon this definition appeared in the general chapter, we were able to develop the monographs. And the next actually component of the monograph is the definition. In definition, we present the content of the dosage form and the limits, acceptance criteria for the limits. In the USA, dietary supplements are expected to meet 100% label claim through the declared shelf life under recommended storage condition. And the formulation of gummy products pose additional challenges compared to the tablets or capsule preparation due to dietary ingredient stability issue. Because it's the matrix is contain water and low pH, which affect stability of many dietary ingredients. And due to stability issue and manufacturing process, manufacturers regularly add an extra amount of the nutrients during manufacturing to compensate the loss during storage in order to achieve the declared shelf life and still have 100% of labeled claim for each ingredient. And establishing specification for the upper limit of dietary ingredient in chewable gels is challenging. Because the dietary supplements with a wide range of doses are on the market, and overage could be a safety concern. Here is the example of two labels, from different gummy products. And you can see that the content of ingredient is completely different. For example, one product has vitamin A with daily value 133%, and another product can have vitamin A only 32% daily value. Folic acid, for example, one product has 100% daily value, and another product 166% daily value. And therefore, manufacturers should be very careful when taking into consideration USP upper limits, which were developed based on the data available to us from different manufacturers, stability data for each ingredient. And because for some doses, its maximum level could be acceptable, but for higher dose, it can be, exceed upper tolerable limit. Here, an example of acceptance criteria for the same ingredient in our chewable gels monograph compared to tablets and capsules monograph. So you can see that based on, again, this specification was established based on the stability data presented from several manufacturers to us. So, and even for single ingredient, the stability in chewable gels are lower and required higher overages in order to maintain the suitable level of ingredient through the shelf life. Here, example of the same ingredient from multivitamins dosage form, tablets and capsules compared to the chewable gels. There are also some ingredient is relatively stable. However, some ingredient completely unstable in chewable gels matrix. For example, pantothenic acid. Based on the data what we receive from manufacturers, we found that manufacturers put up to more than 600% overage for pantothenic acid because it's completely unstable. For this reason, our expert committee recommended to remove the pantothenic acid from the definition for multivitamin chewable gels due to inappropriate stability. So this chewable gel cannot be used for pantothenic acid as delivery system. Another quite unstable ingredient and very important ingredient for dietary supplements is folic acid. Folic acid is found, some company we found use even 400% of overdose, which in many cases, more than our tolerable limit. So therefore, we produce quite high acceptance criteria for folic acid for upper limit. However, our expert committee requested to put particular message for this ingredient that any overage should not exceed the tolerable upper intake level. Another component is the strength, or assay procedure. We spent a lot of time to find out the good sample preparation for gummy product in order to get less variable assay results. And we found that grinding frozen samples is the best way in order to get stable and reproducible results. Therefore, all our monograph recommend cryogenic sample preparation just in order to take a representative sample. In order then it can be different sample preparation like extraction and dependent from the ingredient, but the grinding frozen gummy is what we proposed because it was shown to be suitable for reproducible results. Another important parameter is performance test, and USP monograph recommend dissolution test for all gummy products. Because due to instability, most chewable gel product contains stabilized form of dietary ingredient, including coating to protect some dietary ingredient from the degradation or possible interaction. And protective coating can affect the release of nutrients. Also, gelatin aging may impair the release of nutrients from the matrix. In the table, I showed some examples of assay results, vitamin A permutate, and dissolution results in multivitamin chewable gels as percentage of label claim. And we analyzed a lot of chewable gels on the market, but here just example of 3 products, which shows that not all the products can release, not close to completely amount of, for example, vitamin A from chewable gel matrix. For this reason, it's confirmed that dissolution testing is suitable quality control tool for chewable gels. Another performance test is weight variation. And we recommend to meet the requirements of General Chapter 2091. Quite a long time, manufacturers claim that it is difficult to make it weight variation because of molding and because it's a big variation. However, we did our own investigation, so we bought a lot of products from the markets and make more than 1,000 weights measurements, different lots from different manufacturers, and actually we found that weight variation is relatively tight. Our specification, it looks like tight for chewable gels. However, this specification was developed based on the comprehensive statistical analysis of almost 1,000 weight measurements. Very important specific test for chewable gels. This is water activity and pH. We don't have this specific test for conventional dosage form, but for chewable gels, this is extremely important specific parameter of quality, which allowed to have recommended stability through the shelf life. And we recommend water activity, USP recommend water activity not more than 0.75. However, this parameter may not be appropriate without a controlled pH value, which is not more than 4.5. And pH is another specific test, which is also important factor for quality chewable gel product, and we recommend pH value of not more than 4.5. But again, I would like to highlight that the effect of water activity and pH should be combined to control microorganisms more effectively. Each separate this, if it's pH 4.5 but different water activity, these parameters cannot work relatively well for quality assurance. And a couple of thoughts about the potential safety issue. It is actually general concern that chewable gel finished dosage form may have potential safety concern because of risk of accidental overdose due to its attractive candy appearance and pleasant taste. And eating dietary supplements chewable gels like candy is a common problem. Nearly 50,000 instances of vitamin toxicity from dietary supplements are reported annually to the American Association of Poison Control Centers. Most reported issue for iron overdose in kids and fat-soluble vitamins A, D, K overdose in adults. Also, nutrient overreach is not reported on the label, and for this reason, label actually mislead consumers about the amount of nutrient they consume. And in my final slides, I would like to provide some recommendations which we consider important to take into consideration when manufacturing chewable gels. Manufacture for dietary supplements, and manufacturing should consider the risk of adverse effect from unknown degradation products, as well as the uncertainty associated with the presence of a huge amount of degraded vitamins or botanical, because in dietary supplements we don't investigate this. A stabilization process used should not compromise the ability of the chewable gel dosage form to release the dietary ingredient for potential absorption. Also, manufacturers should consider established safe tolerable level for each ingredient, and try to not exceed this level even if overage are used. And make sure that the manufacturer's product maintains its safety and ability to deliver the labeled nutritional ingredient for potential absorption through its expected shelf life. So it was my last slide, and thank you very much for your attention.
Thank you again, Natalia, for your presentation. Our next speaker is Mr. David Tisi, Technical Director at Senopsys LLC. The title of his presentation is Drug Product Palatability.
Thank you, Brandon. My background, I'm David Tisi, the Technical Director at Senopsys. I'm a food and sensory scientist by training. And when I told my kids that I was going to come to Washington, D.C. to talk about candy, they were so excited they wanted to practically jump into my luggage. So hopefully everybody else had that too. I wanted to talk about palatability in drugs and flavor, really starting with some definitions. You can look to the dictionary to find definitions of palatability. Those aren't particularly applicable to drug products. You can the agencies have definitions that are not actionable as well. So really, we're fighting for those definitions. But in terms of flavor, to a sensory scientist, it's important to define flavor, and for a sensory scientist, it's everything that's perceived in the oral cavity as a product is taken into the oral cavity. You should be thinking about each of these as today's presentations go on, because flavor is comprised of 4 different areas. First, starting with basic taste, this is the perception of molecules dissolved in saliva on the tongue and in the oral cavity by taste receptor cells, also known as gustation. You're limited to 5: sweet, sour, salty, bitter, and umami. Next is olfaction, the perception of volatile chemicals in the sinus cavity by olfactory neurons that are located there. The products can get there through either orthonasal or retronasal olfaction, which is reaching those receptors either through the headspace or after mastication. But again, these are 2 completely separate areas of flavor. You have to think about taste separate from aroma, as different as sight is from sound. They are processed by different receptors, thought about in different points of the brain, and are completely separate from each other. It's just that we're thinking about these together in terms of flavor. The third element of flavor is known as feeling factors or chemesthesis, or trigeminal irritation, depending on where it's located. This perception, chemesthesis, is the perception of really the stimulation of thermoreceptors located in the epithelium that are there generally to measure the temperature change of the oral environment. But in the case of chemesthesis, you're triggering those receptors via chemical stimulation and not a physical stimulation. So you can see that there's a number of different, all different temperatures can be triggered physically as well as chemically. To your brain, that signal is processed in the exact same way. So when you describe a chili pepper as being hot, that is a true statement as far as your brain is concerned. It's just that one way you're getting it physically, one way you're getting it chemically. And then the final element of flavor is texture. It's how a product deforms upon mastication. We as adults eat 3 times a day, so we're very adept at chewing and how this product deforms. Pediatrics that have not learned to chew products, they have a very different interpretation of what texture is and how it comes along. But all of those need to be considered and each are very appropriate in terms of palatability and what a formulator has to deal with. Moving on to what do drugs taste like? Well, this is some data. These are 150 new chemical entities, so this is not OTC data, but new chemical entities that is really all over the map in terms of what are the aversive attributes that formulators have to deal with. On the left side, you're looking at the primary attributes. These are, we'll call it 70% are basic taste bitter as the primary challenge. But when you delve to a separate layer of that on the right, it really is all over the map. These products, just the active itself is bitter and it has an off aromatic, or it's bitter and it has trigeminal irritation. So each of these, again, flavor is a multifaceted multifaceted challenge. And that is not just the case for NCEs. It is the case, of course, for OTC monograph products. Here's some examples, not meant to be exhaustive by any means, but you take something like ibuprofen, which has a bitter taste, and it has a burning mouthfeel, and it has a characteristic aroma that's part of it. So all of these have to be thought of and dealt with together by the formulators that are putting these products together. The world of sensory science, how you can objectively measure flavor, is really divided into two separate buckets. One are analytical methods that use highly trained individuals that measure the intensity of, quantitatively measure the intensity of attributes that are present, any of those elements of flavor, versus affective methods. This is the Pepsi challenge. This is consumer testing that says like or dislike. You use that to make marketing decisions, but the development is really left to analytical methods due to its quantitative nature. These methods are published in various journals so nothing really is proprietary here. As well as inter-industry organizations. So some of these methods, like the ASTM, American Society of Testing Materials method of the flavor profile method, measure what flavor is, and it does this by identifying, number one, what are those aversive attributes, which are the attributes that are present. This could be any element of those flavors, basic tastes, aromas, feeling factors, and mouthfeels, and measuring their intensity measure their intensity on a calibrated scale. Again, these are using highly trained individuals that measure the intensity of what there is, what is present there, calibrating much like you would calibrate a pH meter. This is how the food industry does it. This is how the drug industry does it in terms of defining what is present in a formulation. And importantly, you're measuring these in the aftertaste as well, not just initially. Many drug products linger for a long period of time. You might ask where that scale came from and why doesn't this go to infinity? The answer is you reach receptor plateau. This is really the fundamental graph of the field of psychophysics, with the perceived intensity on the y-axis and the concentration of the stimuli on the x-axis. That establish a sigmoidal scale. Importantly, as you march up through the sigmoidal scale, you reach a couple of thresholds. What is known as a detection threshold. This is the point at which the signal-to-noise ratio breaks through. You can tell that something is present there, but you cannot understand exactly what is being perceived. This is in the field of acoustics. This is you hear a noise down the hall, but you can't understand what's being said. You then reach a recognition threshold. This is the point where that noise becomes a whisper and actually language is translated. And then you reach receptor saturation at the high end. So what does that data look like? So this is an example of a flavor profile of an OTC product. This is how an analytical sensory scientist breaks down what is present in a formulation. Just like an HPLC would break down what is present in the chemically. So what is present here is basic taste sweet, then sour, then those positive aromatics, and then you get some negative aromatics and bitterness thrown into there. And of course, you're measuring these in the aftertaste. So this very much is a quantitative measurement of what a product tastes like. As I mentioned, a sensory scientist looks at the output of a flavor profile just like an analytical chemist would look at the output of a chromatogram. This is illustrative, by the way. But they're looking at the individual peaks as the individual attributes that are present, the peak height or AUC as the intensity of those attributes, and how long those persist in the aftertaste as the elution time. So again, just a very quantitative method of figuring out what the flavor of these products is. So some examples of OTC actives at their clinical strength and how they persist in the aftertaste. Some go out for a long period of time. And again, something like ibuprofen, basic taste bitterness, yes, it's bitter, but that's not its primary challenge. If I showed you the burning graph, that would go out much higher and look very different. So tying this back to what that threshold, the recognition threshold, the point that as a whisper down the hall becomes language, you're really trying to make a palatable drug product, not necessarily by taking the aversives down to zero. You're trying to take them to the point where a consumer is not saying that this is bitter and starting to have an aversive reaction to it. So again, that recognition threshold, not the detection threshold. To a formulator, what can they do? What are the tools that they have in their pocket to deal with those aversive attributes? Really, there's 5 that a formulator uses, either OTC or NCE. The addition of a flavor system. The addition of a flavor system. Number 2 is the addition of some type of encapsulation. This is either putting a barrier coating, a Worcester coating, or an ion exchange resin, a film coat. The third way you can deal with this is the use of alternative API forms. That could be in the form of a new salt moiety or a prodrug. Again, long development timelines associated with those. Chemical complexation. This is adhering the active drug product to to another molecule that will prevent the perception. Typically, beta-cyclodextrin is used very classically. And then 5 is signal interruption. Really, the early interruption of that perception of bitterness at the tongue or in the signal cascade responsible there. But really, in the field of OTCs, number 1 and number 2 are the ones that are used most typically. So that first one, flavor systems. Really, to a sensory scientist, what is the flavor doing? We are really leveraging the concept of mixture suppression or taste-taste interaction. So what we have here, I don't know what that second box is, but what we have here is the on this first graph. This is some research out of the University of Oregon. This first bar is the intensity of quinine sulfate. So what mixture suppression tells us is that when you add basic taste to other basic tastes, you're bringing down the perception of the target basic taste, the high one. You are blending that away. So if you start with the bitterness of quinine sulfate and then add sucrose, you get a reduction. If you add sucrose and sodium chloride, you get a further reduction. If you add sucrose, sodium chloride, and citric acid, you get even a further reduction, even though these concentrations are there. That's what a formulator is doing with a flavor system. And this holds true for if you have an extremely aversive salty product like a colonoscopy prep. You would also add other basic tastes to drive down the target basic taste. Sweet, very rarely have in my career encountered a sweet drug, but if it was too sweet, you could add those other complementary basic tastes to bring down the perception of sweetness. Same with sourness. Again, taste-taste interaction. You've probably come across this when you were in the kitchen, this concept at a very fundamental level, by making lemonade, right? So you take a very sour lemon juice, and how do you make that less sour? That's your aversive attribute. Well, you're adding, in the case of lemonade, putting in sucrose. Now, is that changing the pH, or is that changing the titratable acidity? Not particularly, but you're really leveraging that sensory practice of mixture suppression. So the different excipients, of course, can be used to get there. Frequently sweeteners are part of that mix. Those would be the same for acidulants and taste modifiers, like salt. So there are many different sweeteners that can be used. You might have seen a table such as this that talks about the relative sweetness of those different sweeteners. Looking at all tied to the relative sweetness of sucrose, which is the things that are thousands of times as sweet. In the case of sucrose, for most concentrations, you have a fairly linear response, whereby you dump in more sugar and it gets more sweet. But the numbers that you see on the table really are an oversimplification for things that have nonlinear responses. Most high-intensity sweeteners have a nonlinear response. Something like sodium saccharin here, it's rated here at 300 times as sweet. that's mostly at these low concentrations because you get that plateau so early on in the concentration. And in fact, something like sodium saccharin or most high-intensity sweeteners have at strong concentrations some bitterness that is brought into that. If you are at a cafe or if you see a pink packet in the atrium outside, if you dump an entire packet of sugar into your mouth, it's going to be fairly pleasant. If you dump an entire pink packet, the sodium saccharin, it's going to be revolting because you have stopped being sweet and you are adding bitterness to that formulation, to your mouth. So not only intensity is important, but how long those persist in the aftertaste, just that different sweeteners have different times that they persist in the aftertaste. Sucrose is the gold standard and has a strong onset and then fades very quickly, but other ones have different time delays. But starting to get to some data, this is what you're going to be seeing on the next couple of slides is an overview of 97 branded pediatric products. These represent the pediatric cold, cough, and flu and analgesic sections, because we're really interested in what does a drug taste like. And this is solution, suspensions, drops, chewable tablets. So what we have here are those, again, the scale goes from 0 to 3, and how strongly each of these are in the or how strongly these are in a histogram form. So what does your average, what does the mode of pediatric drugs look like? So we have, most pediatric drugs have a sweetness that are slightly below a moderate intensity, a bitterness that ranges between a moderate and a slight and a moderate, and aromatics. These are the positive aromatics, the cherry, grape, and orange, that go between right at a mode right around a moderate intensity. So you have to think about, okay, so that's what your average or the world of drugs products tastes like. What is the flavor of those? What does a candy taste like? The world of confections, you can distill it in most broad senses down to about 3 different areas: hard candies or boiled sweets, chewable candies, gels, and chocolate-based confections. So what do each of those taste like? From the hard candy side, you're left with the products that are extremely sweet, have no bitter, and have aromatics that are also particularly strong. In the chewable candies, they have, again, very sweet, no bitterness, maybe a little bit less intensity aromatic. Now, chocolate confections are probably the most disparate here, that they have, they're fairly moderate, they're more moderate in terms of sweetness. They do have some bitterness, and the aromatics are slightly reduced compared to those other sweet types. So if you're really comparing what a drug tastes like to what a candy tastes like, drugs are lower in sweet, or at least OTC drugs are lower in sweet, as on the whole. They are higher in bitter on the whole, and they are lower in aromatics as on a whole. So there are differences in the perception of flavor between drugs and candies. And that doesn't just stop at those intensities. That also is talking about, I didn't even talk about sourness. Sourness, some confections, warheads, Sour Patch Kids, they're extremely sour, almost to the point of novelty seeking. That is very different between drugs and candies. Texture. I can't really harp on this enough. The fact that almost all confection products have a novel texture. A hard candy, you were trying to replicate everlasting gobstoppers, have that sticking around in the oral cavity as long as possible. Chewable candies really play into the novelty effect of chewing a product, having it break down, squeeze between your molars, and reposition it. There's novelty in there. The most impressive is probably chocolate and chocolate-based confections. You don't really think about why do people talk about chocolate? They love to talk about the aroma. Really, it's the texture. Cocoa butter has a very sharp melting point right at the point of body temperature. So you place a hard candy, a hard chocolate confection in your mouth, it becomes a liquid, and there you have immediate release of the aromatics and the aromatics and sweetness that is there. We love chocolate so much, you can take out the flavor, and it becomes an enjoyable product. If you've ever had white chocolate, you have taken the cocoa flavor out of that product, and it becomes a palatable formulation. You're eating plant lard, but we love that change. We love that texture change so much that that's what we perceive. Obviously, major differences in branding, major differences in the packaging and the color intensity. Extremely vibrant colors. Again, you're just not seeing most of these areas in the field of OTCs. You are in every commercial candy product. Color has a big change. Color has an effect on the perceived flavor intensity. So as you increase the color, that flavor intensity goes up, as does overall acceptability. So most confectionery companies are very interested in pumping up the color because of those responses. And of course, these questions that I brought up are not going to be limited to the more simple route of oral liquids, like in most OTCs. When these actives are placed into gummies, films, chocolates, you're going to have the same aversive attributes and the same need to cover those up. So really, the question is, have non-prescription drugs been developed specifically for palatability or to promote liking? And you can see that there's a difference in their overall perception, so they're not exactly the same. But of course, it's a balance on between rejection and overdose that we're going to be talking a lot about today. Thank you. I just wanted to mention this. Fran from my panel, I asked what should we call this formulation, this which I called my talk, and she talked about, well, trick or treat has two meanings. I said, what do you mean? And she said, well, there's treat as in to cure, and treat as in a delicious substance. So I thought that was very interesting that treat has both those definitions that are appropriate here. And thank you very much.
Thank you again, David, for your presentation. Our next speaker is Dr. Xiaoling Li, Professor of Pharmaceutics from the Thomas J. Long School of Pharmacy at the University of the Pacific in California. Dr. Li's presentation is titled 3D Printing Technologies for Oral Drug Delivery.
Well, first of all, I would like to thank Brendan and Kevin for inviting me to give this presentation on the topic I'm very passionate about and have devoting quite a few time onto it in the past 8 years. Since some of the content I'm going to talk about are related to a company I founded 8 years ago, so I'm obligated to have this disclosure. To put everyone on the same page, let me start with what is a 3D printing. 3D printing actually is a digitized process using the computer-aided design to create object on the computer, whatever shape or geometric structure you would like to have, you will be able to create on the computer, and then we'll convert that structure into an instruction we'll be able to execute by the computer to instruct the printer to print the object layer by layer. So basically, there are several mechanisms you can use to build this layer by layer structure. The first one is using the polymerization that will involve the chemical reaction. And then we can start to play with the temperature. You increase the temperature, make the material flowable, and then you cool it down, make it solidify. And you can also have a whole bunch of small particles and then put an adhesive, bind this small particle together in a layer by layer manner. The last one is going to be the extrusion or ejecting, and again, that would be used to build each layer. In the in the biomedical science area, this 3D printing has been used in various areas, so as I show in the slides. But today, what we'd really like to talk about is the pharmaceutical products, how we use the 3D printing technology in this area. Specifically, I would like to talk about three aspects. The first one is the personalized dosing. The second one is the drug delivery. The third one is the manufacturing, how we can utilize the 3D printing technology to achieve all these three aspects. To give you a little bit background about the player and the status of the 3D pharmaceutical 3D printing technology in the pharmaceutical area, I put up a little bit history. To start with, it's when the 3D printing technology is invented. It's in 80s by Japanese. And then in the pharmaceutical area, the first appearance is in 1996 by Dr. Sima in MIT. And he used this powder binding technology to create a solid dosage form. And a year later, a company formed, licensed this technology called Theresa. And unfortunately, Theresa convert their interest into the medical device, but some of the hardcore pharmaceutical scientists spin out a company called Aprecia. The Aprecia later launched the first product in 2015. So we do have one 3D printed pharmaceutical product in the market right now, approved by FDA. The personalized dosing representative would be FabRx, which is a UK company. The company I'm involved is in extrusion-based 3D printing technology. So we have powder binding, extrusion-based, and various technology used for personalized dosing. From ASTM point of view, the 3D printing technology could be divided into 7 categories. And among these 7 categories, those big check marks would be the primary application of 3D printing methods in the pharmaceutical area. In the literature, one of the methods, which is extrusion-based, it's called FDM, has been extensively published and studied because it is a very simple machine and easy to acquire, low cost. So we have using this type of technology to create oral tablets, capsules, or even implantable device and dermal patch like a microneedle. Let's first take a look about the individualized dosing or personalized dosing. This is a very good concept, but regulatory-wise, I think still it's not really mature. Okay, FabRx in UK is the company really trying to push this concept moving forward. The application in the cycle, I think, is quite obvious. We can see a lot of benefit out of it, right? They have used this printer created by themselves. It's basically FDM-based, okay? What FDM does is you make the material into a filament, and then let the filament going through a printing nozzle, and then melt, and then build layer by layer. And they have different type of the color or shape, and try to test the acceptance of the patient and its efficacy. Basically, they want to see how 3D printing technology will influence patients' acceptance. And this is the first product approved by FDA. It's called Spritum, and the technology is powder binding technology. Basically, what you do is you have the powder making into one layer, and then selectively spray the adhesive to bind some of the particles together. You put another layer, you bind them, so slowly you will build a tablet. The characteristics of this product is it can instantly disintegrate, so it's very fast. utilizing the 3D printing technology, really what we can do or what we can achieve is try to build a structure, both internal structure and external structure. So external structure is more appearance-oriented, but internal structure will offer a lot of advantage to have a different mechanism of the release or mode. So that lead to the technology I'm involved. It's called melt extrusion deposition 3D printing technology. In this technology, what we do is try to continuously convert the powder material into molten or flowable material and then build the object layer by layer. So this printer head can be precisely deliver very small amount of the molten material and then build the object layer by layer. So compared to the FDM, we don't really need to make the filament, and that would take a lot of restriction out of what kind of material we can use, what kind of API we can incorporate. So if we have multiple printers coordinate with each other, in this case, let's say we have 3 different printer heads and handling 3 different type of material, we can build a very complicated structure. For example, we can have coat and shell with a coating or with a seal on the top, and they can serve different function, right? We can build different shapes, and we can build different compartments. And these different compartments can have a different API or same API. So essentially what we did is from the software and hardware point of view, create a technology or instrumentation specifically for pharmaceutical application. And the material we're using is all pharmaceutical excipient or glass material. So we took about 206 glass material and pharmaceutical excipient and built a database. So we have a good understanding about its melting point, softening point, maximum daily allowable amount, and melting point, etc. Okay, flowability. And coupled with that, we also have the structure design for different dosage form. So let me quickly talk about this structure. Why do we want to talk about it, and why it is so interesting for us? Let me just take one example to say if I have a 3-compartment tablet, which you will be able to control the components in there or API in there individually, and we can put 3 different excipient in there, and then you can have 1 immediate release, 1 zero order release, and 1 delayed release. And I keep using this example in my talk, saying we can have a first compartment containing zolpidem. If it's going to knock you out right away, right? It's a fast onset, but it will not last too long because the half-lives are very low. So we can put the second compartment containing melatonin to maintain your sleep over the night. But in the morning, you really need to wake up. So we can put a third compartment using caffeine. So you can wake up and have a normal day, right? So those would be what a multiple compartment can do for different API. But later I will give you another example to talk about with the same API, we can also do a lot of things with the individual compartment. We can use different materials. So here are the examples. We can use the sugar-based material to build, again, different compartment. And here's one of example, and the structures look like this. So what we did is one of our kind of like a demo tablet. Okay, we have 2 compartments. One compartment will have apple flavor. The other one will have orange flavor, but they come at a different time. So when people take this tablet in the mouth, first you're going to sense apple flavor, and then later you're going to have orange flavor in 2 or 3 minutes span. So how do we do it? Well, we put them into different compartments and let them release at a different time, okay? So essentially what we did is have this cover of that compartment using different material, and we can precisely deposit that material onto the surface and make it as a seal. Well, in addition to, multiple compartment, what else can we do? There are many pharmacokinetics profile will have different clinical application, right? So you may need to have something which has a zero-order release, and we can just have this tablet built with a shell and cord structure because it's layer by layer. So you can build each layer with the same surface area. So each layer is going to come up the same amount, right? But we can change the surface area of each layer. For example, in this case, you're going to have small surface area to start with and then gradually become bigger, so your amount of release at the time is going to increase. And you can have it slow, fast, and then slow down later, and you can have a loading dose at the very beginning and a very quick release rate. By the way, if you look at the slope, you can see the release rate. And then you can slow down, and then toward the end, probably absorption is not that great. You won't have more coming out, right? And it will accommodate our GI tract. So here you can see the theoretical model is one of the line, and experimental value is another line. And all these is mathematically described. So you can see, the prediction is very good, right? In mid-70s, for those in the pharmaceutical area, probably most of you have heard Higuchi's name. This is younger Higuchi, Bill Higuchi, and his one of his students, Bob Lipper. He later become VP of Scripps, but now it's like a BMS, right? It's a part of the S is a Scripps. They proposed a geometric shape which will be able to control the drug release and offer a zero-order release rate. But for decades, no one can really achieve this because it has a very special geometric shape. With the 3D printing technology, using the MET 3D printing technology, we will be able to build a tablet with that compartment, with that specific geometric shape. And we further get into this model and then build to study each parameter, the opening, the angle, the depth, and see how they will influence the release rate. So we have a wide spectrum of the release kinetics we can cover, just use one of the geometric shape. We can build other structures. For example, we can build a structure with a delay release mechanism. We have core and shell, and by varying the thickness of the seal, we will be able to determine when this drug will start to release. And you can even have a zero-order release built into the core, so you can have delay and kinetics release control. And we can have multiple components in there. In this case, we have two API. One is in the orange color, and then the other one is in this teal color. So the teal color is going to be zero-order release, and the orange color is going to be multiple pulsatile release. So in vitro, you're going to see the teal color is a straight line, right? Zero-order release, but the orange color, you have first pulse, nothing happened, and then the second pulse. And in the dog study, you can see, definitely you're going to have constant release, PK profile, and pulsatile PK profile. By utilizing the multiple compartment, we can easily modulate the PK. And in this case, in this application case, we call Lego approach. So what we do is we have one compartment using the immediate release formulation, and the other compartment use extended release formulation. And first, we will put these two formulations into the dog study to get animal or human and get a parent PK. So we harvest those PK parameters, and then we will do a simulation by varying the ratio of these two formulations. We will be able to get the target PK profile. So with that, you can see, based on the simulation, we can achieve that target just in one formulation attempt. So with all the example I just give you, I think I probably already give you an impression. The 3D printing technology could help us to make the formulation development much more predictable. So we developed a platform called Formulation by Design versus the current practice. Most of the formulations are formulation by trial. Okay, try and error. Finally, you're going to get to that formulation. So with this approach, we'll be able to make a lot of things much more predictable. So this 3D printing formulation by design approach would start from the PK target PK formulation, and then convert it to in vitro release profile, and then I think my time is almost up. I need to speed up. And then we pick the model, and from the material database, we will pick the material to build this structure, and then using the 3D printing instrumentation to achieve the structure, and then have the in vitro and in vivo release. So instrumentation-wise, we built two types of instrumentation. One is for R&D or early clinical development, which is a smaller machine. And then we have 3D printing system, which is for full-scale commercial production. So the production line basically divided into three zones. The first one is material preparation zone, printing zone, and then packaging zone. So the whole thing would be automatic. So I don't know if I can get this one going. Okay, yeah. So this is a small machine, and you can print one tablet at a time or print 4 tablets at a time. So as you can see, start from modeling and then handling different material using different printer head. And in this case, it's a 4 tablet printing at the same time. And each layer we print is going to go through a laser scanner to measure the height. And this is the production line. And material will come from the preparation zone. This is a material preparation. And then using this robotic mobile vehicle, put it into the printing zone. And after you get into the printing zone, it has a 32 printer head printing at the same time for each material. So you have multiple stations to handle different material. And after printing, those robotic arm is going to take it and then put into the mobile robot and then put into the packaging zone. So the whole production line is automatic, okay? And each tablet will given a QR code. We can trace all the printing condition for each tablet, okay? So in a way, what we did is we tried to use this by design concept to change the pharmaceutical industry common practice. So started from the drug delivery, it's structured by design. In the product development area, we have formulation by design, and in the manufacturing, it's quality by design, right? So it's a whole thing is by design approach. And what we like to achieve, or we are achieving, is a digital product development and digital manufacturing. So with that, I think I would try to associate what I taught with the 3D, with the candy-like. With a very limited knowledge about what the candy-like product would be, I give some feature or characteristics for the candy-like, and I may not be right because I think a lot of experts are here, and toward the end of the day, I think I'm going to be learning a lot. So limited knowledge, I think, temptation is one of the factors, and then if we create the pressure or not, and if this use of this product will have some dependency or not, and the user control would be another important factor. For example, we have addict, right? It's a lollipop of a fentanyl. It is a candy. It looks like candy. It tastes like candy, but I'm not so sure we should classify it as a candy-like drug because it's highly controlled. It's not freely accessible for the user. So with that, if you look at the left-hand side, you're going to see some of the features we can use to give these products, the candy-like product, with all these features, which, may be very difficult to achieve or may not be able to achieve in the past. And using 3D printing technology, we should be able to achieve it now. Well, I think I want to skip this one because it's ADHD, and we have developed something to reduce the stimulants used and to reduce the dependence. That's one of example. To summarize, I think I would give this slides to cover some of the application of the 3D printing technology for pharmaceuticals. And usually I would say, your imagination is the limit. There are a lot of things we can do using the 3D printing technology, and we just start. And it's a very exciting area. I hope, a lot of people will look into it. And although the example I'm using today, many of them is a prescription drug, but I'm pretty sure the technology can be easily triggered into the OTC or Monographs drug. So with that, I would like to thank my team. Now it's about 140 people. 1/3 of them is engineer. And also would like to thank some of the organization. And on your right-hand side, you see the company received the TCT Healthcare Award. TCT is a 3D printing community. The annual award is considered as an Oscar in 3D printing. So we are very proud of we received that award. So thank you. I think I'm going over my time.
Thanks again, Dr. Lee. So that brings us to our first panel. So in addition to our previous speakers, Dr. Natalia Davydova and Dr. Lee, who will be serving as panelists, I am also happy to introduce Mr. Jeff Worthington, President and Founder of Senopsys. Additionally, I'd like to introduce Dr. Swapan D, Senior Chemist with the Office of Pharmaceutical Quality here at FDA. Our first panel session on formulation considerations for solid oral candy-like dosage forms will conclude at 10:30 a.m. And just as a reminder, if time permits, we will try to take questions from the online Q&A chat. In addition to that, we have a microphone set up at the center of the aisle for our in-person audience. So at that time, again, if time permits, we'll invite you up to the microphone to ask your questions. But with that said, it's also my pleasure to introduce our moderator for this session, Dr. Danai Christodoulou, Branch Chief of the Office of Pharmaceutical Quality here at FDA.
Good morning. The first question we have for our speakers is regarding your definition of a candy-like dosage form. So from your professional perspective, how would you define a candy-like drug product, and what characteristics contribute to this definition? And we can start with Dr. Davydova.
As I have mentioned in my presentation, that we have definition for gummy product, which is like candy-like, and it's available in General Chapter 1151. And partially definition I already presented, and yeah, I can actually I have this general chapter, and I can just briefly read what it's in how USP defined gummy products. So it's under this definition. It came under gel dosage form, and these consider like chewable gels. Again, I mean, it's only like chewable gels. It's much more other candy-like products. These actually it's actually chewable gels are used to deliver drug substances and dietary supplements via the oral route. In addition to the drug substances or dietary supplement, chewable gel can consist of all or some of the following components: gelatin agents, sugar, water, sweeteners, and flavoring agent. The sweeteners and flavoring agent intended to enhance patient acceptance and mask the taste of the delivered labeled drug substances or dietary supplements. Chewable gels maintain their molded shape, are elastic, and yield to mastication. They are intended to be chewed before swallowing. Chewable gels are also known as gummy in the confectionery and dietary supplements industry, but that term is not used in official article title. Also, we have definition for preparation of this finished dosage form. Chewable gels are formulated with one or more gelatin agent, such as gelatinous starch, sugars such as sucrose, fructose, or corn syrup, flavoring agent, sweeteners, colorants, and water. The ingredients are blended and heated to form a viscous solution that is poured into molds, i.e. cornstarch molds. After cooling, the individual units are separated from the molds. This is what we have in USP, and this is nothing more I can add.
Thank you.
And this came after a lengthy discussion with FDA, including Liaison, which participated in our dosage form expert committee, nomenclature expert committee, and dietary supplements manufacturers. So this definition is acceptable by dietary supplements manufacturers for gummies.
Thank you very much. And just continuing with the formulation, I'd like to just pose the same question to Mr. Worthington. If you can add anything from your perspective in what constitutes maybe a candy-like dosage form.
My pleasure. First of all, thank you for FDA for inviting me today, and M-CERSI for organizing the meeting and facilitating travel. Candies are very complex. There's thousands of them. Our children will be coming home with them tomorrow night, and as my colleague David indicated, they differ in all kinds of dimensions. We approach the development in terms of what is necessary to make a product palatable. We don't set out to say, how do we create a candy? We set out to say, how do we create a palatable drug product? And so to do that, as my colleague David presented, unlike most dietary supplements, drug products tend to be extraordinarily bitter or have other aversive tastes. The taste masking challenge is orders of magnitude greater than most dietary supplements. Of course, there'll be the random exception to that statement. So as such, we follow the approach which is really used in the drug industry that FDA would look at for a new drug application for an investigational drug, in that you need to propose every ingredient in the drug product as well as its usage level in terms of what is its functionality. So And from a palatability standpoint, we say, okay, which ingredients are necessary to reduce the aversive bitterness or mouth irritation or whatever it happens to be, and only look to include those excipients that have a measurable impact in the reduction. And as my colleague described, it's taste-taste interaction. So you add sweet to reduce bitterness. By doing that, you're reducing the perceived sweetness. So you come to the point where you have reduced the amount of bitterness to the greatest extent possible. Adding more sweetener has no positive benefit. So it's really a propose and justify from building the formulation from the ground up. Same goes for the flavoring aromatics. The cherry, orange, and grape are typically you add them to get to the point of patient recognition. You're not trying to create more flavor, as in terms of the aroma, than is really necessary unless you have an aroma masking challenge. And then texture, it's the wild, wild west of textures. That's what makes candies so wonderful. From our standpoint, we typically view texture as, do you need it to deliver a sustained release, as in through chewing, as an example, or by building viscosity to suspend the active. So our view is we create a drug, not a candy, nothing more.
Thank you. And Dr. Lee, you touched upon shape and the stability of the shape and how that is formed and what happens after chewing. Can you also tell us from your perspective what do you think a candy-like dosage form could be defined as?
From dosage form point of view, I think immediately probably would think about lozenger, like a pastille or choky. But I would like to think if we want to define something as candy-like, there is also a psychological factor.
Psychological factors,
yeah.
If
the patient or user, not necessarily patient, really want to come back to have more. Okay, so I think that's part of feature of a candy, right? The kids get the candy, they get sweetness, they like to have more. So that should be another factor to be considered. Although the topic probably will be more in the area of the OTC or monograph, but some of the prescription drug probably have abuse tendency, could be in that category too. So that brings to another issue is the control, accessibility. So if we have, the external shape or the taste is one factor, and the other extreme probably would be the accessibility. And in between probably would be the psychological factor.
Thank you.
And of course, the dosage form could be able to contribute to all these factors. Yeah.
Yes. Dr. Sobande, would you like to comment?
Yeah, may I ask you some question? I think this is very interesting, this 3D technology, because the drug is forming layer by layer, right? So is that the excipient solutions and the active solutions? Do the excipient solution they are separate, or each excipient is forming a layer?
Yeah, we can, depends on the design. We can have a layer of excipient, a layer of the API containing formulation, and we can also just have the homogeneous one. So it really depends on the release mode, or what will be our target profile. For example, you want to have a mini pulse, then we can just create what you just described, as a very small interval and one layer, and then just very short period of delay, another layer. And we can also just have all layers.
My next question is, since we are talking about candy, do you see any issues if you use sugar or that type of excipient in this formulation?
No, actually, the sugar is one big category we're using, especially for oral cavity dosage form, like mannitol, sucrose, mannitol, all those polyols. We study them and understand how fast they would dissolve and what is the soften point, melting point. So we have a good study about this material, and it really depends on the application. And if we want to have an application with a relatively slow dissolution, it will be similar to the hard candy, right? So if we want to have something dissolve much faster, probably will make a more porous structure than...
I'm sorry, just to quickly interrupt. We're having some trouble with our online participants hearing. So if you could speak closer to the mic, that would be great.
Okay,
thank you.
Thank you. So it really depends on the structure, and that is the advantage of using 3D printing. For example, if we want to, using the sugar-based product, if we want to make a oral disintegrate product, we can have hard candy build a ring, which will hold the structure, and inside you can have very thin threads. So it would be just like a cotton candy. So when you put it into the mouth, that cotton candy is going to melt instantly.
So you think that that chewable gels or gummies-like product can be created by this technology?
Yeah, but just from the manufacturing point of view, if it's a gummy, I'd probably go for molding instead of 3D printing because it doesn't have a lot of structure requirement. Yeah.
Thank you. So moving on to the second question, can a drug candy-like dosage form be made such that is very distinct from candy? For example, there are regulations about debossing, embossing, imprinting, and is this possible to be achieved on the candy-like dosage forms, just as we would have it in a capsule or tablet? So what would be some distinction factors from a candy-like dosage form with a candy? How can we distinguish these two if both were to be on the market? Go ahead, please.
Just again, I mean, I just would like to share just what situation with the dietary supplements. If we take, for example, very popular gummy bear for children, so we have actually you can distinguish them in the store because they have separate packaging. So it's in bottle dietary supplements and candy have different packaging, and also they label the dietary supplements and candy. However, and they are located in different places in the store. However, when you come home, it can be stay on the same shelf, for example, for children. But if you take them out from container, they are not different at all. And here's the problem that is possible overreach if the children have access to the dietary supplements, because if they came from school, they are hungry, they just take candy, and then candy die, okay, I will take 2 dietary supplements, okay, then 2, and then 2, and then it's possible overages. From actually possibility of the printing, of course, I mean, what it's we saw on the market, you cannot maybe use exactly that technology of the printing, but some printing on the chewable gels is possible, because we saw very advanced, for example, chewable gels which contain, for example, different portraits on the surface. So which I can conclude that some sort of printing with notification actually for drug it exists. Actually, they can hold and be visible. So based on the dietary supplements, what is available on the market.
So we have some possible imprinting on these chewable gels.
I believe so, based on what we saw on the market.
Thank you, Mr. Wasserman.
Yes, I'd kind of like to take a little bit of a different tack. You know, hear a lot about gummies so far today, and probably more so. My viewpoint is it's a technology looking for a solution. What is it that gummies would deliver for a drug active? Are they providing something that a medicated gum wouldn't, that a solution wouldn't, or is there an effective release from the chew characteristics that's unique and different from an orally disintegrating tablet, from a chewable tablet? I think there's lots of things that I can point to of a gummy that I would rather not see in a drug product. I don't think the shapes should be consistent. Things like rings and worms, cartoon characters, et cetera, and a lot of the iconography that goes along with gummies and a lot of other dietary supplements. There's a reason that dietary supplements have a different regulatory framework than drugs do, and I think we should recognize and appreciate and adhere to those boundaries. So you know what a candy is when you look like it. It may be very hard to define, but I would advise or just recommend that we kind of continue we're trying to deliver a drug active in a safe and efficacious manner. And some of these forms are a means of delivery, not a means of delight. Thank you.
Thank you. So you're suggesting that some boundaries, if we set some boundaries in shapes.
Absolutely.
Okay.
I would not allow, as I indicated, shapes. I would eliminate lentil shapes. So a lentil looks like most candies. Probably not supposed to mention names, but they look like M&Ms, Reese's, et cetera. So I would not allow that shape. Given my druthers, I would not allow, characters and anything, stars, diamonds. That's not the purpose of a drug product. I would not have drug products in bags, et cetera. So I think there's clear ways for us to define what the boundaries are, even if we can't actually define what a candy is. I think we might be better served defining what a candy is not.
Thank you. And Dr. Lee, did you have a comment?
Yeah, I think I agree with what Jeffrey said. It's really, there are two steps. The first step is we need to define what is candy-like drug, what belongs to the candy-like drug. And then we can, probably from regulatory point of view, you will be able to give some definition, and then some of the shapes, those attractive factors may not be used for the drug.
And is the 3D printing technology amenable to some shapes that may not be confused with candy?
Oh, definitely. From technology-wise, I don't think that it's an issue. The 3D printing technology has a big advantage. It's not mold-related. It's, if you can imagine it, you can draw it, you will be able to make it. So any shape, as long as you define, or you can draw it, you can even take a picture and then convert it, digitalize, and then using the conversion, making into the machine to print that shape, you just imagine or draw. Yeah. There's no barrier in that.
There is no barrier. Yeah. Okay. Saban, do you have any comments?
Since since this is a manufacturing ideas, I think it would be probably very beneficial. All those things are explained, but how about just the look of a drug product? It may be candy, chewable gels, gummies, even hard lozenges. I understand, based on my experience, I think manufacturing of this type of dosage form is challenging. The reason is it is done in many different ways, either by molding or by direct compression. But the point is, when you directly compress this gummy, gum powder, then the dosage form can come up in between hard lozenges and chewable gel. So how do you control this type of dosage form? And then the applying to the pediatric and geriatric patients, for the palatability and taste, I think one of the things can come to my mind that is it possible to make the shape and color in such a way that it doesn't look like candy, but the taste will be like that, and something moderate. I know this is challenging, but this is the things probably we need to think about.
Great. So we can just, yes.
Can I add an item on?
You have a follow-up comment?
Yes.
Please go ahead.
In terms of color, for most in the prescription drug product, because of color regulations internationally, it's extraordinarily complex. The tendency is to avoid the use of colors. The opposite is true for candies. You can't make them more intensely colored. I would advocate no colors for OTCs.
That's actually a very good comment, because there are some there is a CFR regulation about the colors that can be used in drugs, and colorants are different than other excipients, so we can take that into consideration as well. And this actually brings us to manufacturing challenges for candy-like dosage forms. And we talked a lot about the sugar content. Excipients such as a mixture of glycerin gelatin may affect the bioavailability of drug products. Could you comment on some technical challenges on the manufacturing of the dosage forms, and just give us your own perspective.
I can talk only from what we can see on dietary supplements, and we can see the stability issue. And the manufacturers put a lot of overages in order to keep, in order to keep a reasonable shelf life, because they try to put at least 2-3 years. And from logistic point of view, 2 years maybe not quite suitable, because it can be stay on the shelf completely a short time. So the manufacturer tried to go with like 3 years shelf life. Expiration date. From manufacturer to the end of shelf life. So it's ingredients because of particularly only I'm talking about chewable gels, because of high moisture and acid pH. It is not many ingredients is there. For this reason, the many ingredients in chewable gels are protected. They use protected coatings in order to stabilize the ingredient. But then it's another problem, that because it's some coatings are like a rock and they cannot release a dietary ingredient at all. Again, different from the from the drugs, dietary supplements do not have any clinical study. So we cannot have any pharmacokinetic profile, and in order to say for this reason we just recommend dissolution as a must quality control test in order to at least to show that ingredient can be released even you use stabilizer and to be available for potential absorption. So this is two problem. I mean, it's stabilization, which can affect release in dietary ingredient or maybe drug ingredient, and overage, because in drug you have to keep 100% of the formulate the product 100% of ingredient, the dietary compound. So I mean, it can be challenges maybe because I mean it's difficult to formulate 100% without overages due to stability issue. Maybe this dosage form, it can be only suitable for certain drugs.
For certain drugs.
I mean, it's maybe even very limited.
Thank you. Yes, please.
Yeah, manufacturing is way outside my wheelhouse, but I would just build up on one thing that was just mentioned in terms of the flavorings. This is the aromatics, the grape orange, et cetera. Those are typically complex mixtures of aroma chemicals, and they're the more labile ones, so they decrease upon storage stability. So typically you put more in initially so that you have some flavor left at the end of the expiry. And there's generally no analytical methods. You can't use accelerated testing because they're labile molecules and they degrade.
That's very interesting. Any comments from you, Dr. Lee?
Probably I should just focus on the topic I talk about, 3D printing technology. I don't think it would be a very challenging issue, either using the MET 3D printing technology I'm talking about. That's solvent-free, so usually you are not going to have that high content of water. And also if you use precious technology, it's powder binding base, so start from dry powder. The water content are also very low, and their platform will be able to create a system where instantly disintegrate in the mouth. So the material or the formulation we're using probably is quite different from traditional gummy type of the formulation. The gummy type of formulation, probably one of the important ingredients is gelatin, right? So gelatin, if you have the water content below 10%, it becomes quite hard. The hard gelatin capsule probably is 6 to 10% of the water content, and that's already, kind of hard. And if you want to have a chewy feeling, then your water content got to increase. In my opinion, probably that water content is really the problem of causing the stability issue.
Yeah. Okay. So I think that, shall I go into the one more question for the panel? It's actually a very quick question. If you think there are differences in methodologies involved in assessing palatability in children versus adults, maybe, do you have any comments?
Sure, I'd be happy to address that. I think my colleague David included that in his presentation. Really, there were the two types of analysis methods: the analytical, which is to measure the product, and what are called affective tests, which measures human response to the product. And it's things like liking and preference. For children, you can't use them to develop the product because they can't give you the language to describe what's wrong with it. So really, both from a development perspective, it doesn't matter whether it's adults or children. If you're using effective methods such as I like it, or yuck, or yum, or smiley face, et cetera, it's not helpful for development. But typically for children, you have to use liking with a facial recognition scale. With adults, you have a bit more flexibility. But we all have the same perception, It's just that we don't have the language skills to be able to describe it.
Thank you. So do we have time for one question from the audience in the room? Is there any question? Yes? Can you come to the mic, please?
Duffy Mackay, CHPA. I noticed that does dissolution include mastication when you're trying to figure out if a candy-like form is absorbed? I noticed the USP monograph does not.
So actually, all our monographs, as I mentioned, recommend dissolution testing for quality control, because we found that, first of all, what we found that many ingredients, for example, cannot release from chewable gels, like similar, using the same dissolution procedure as for tablets or capsules for the same ingredient. We have to develop specific dissolution procedure for the same ingredient in chewable gels, because they, especially, for example, vitamin D, the formulation, the chewable gel formulators, that vitamin D practically cannot release, so it's methylated, so you need to develop specific surfactant, specific condition in order to suspend or dissolve the vitamin D in dissolution media. So, and because of stabilization, a lot of ingredients that just use stabilizing coating. So, and again, for this reason, we analyzed a lot of products from the market in order to show that they are not consistent. Some chewable gel can release ingredient practically the same like assay area. Our specification is 75% from label claim. So, but usually when we analyze assay procedure, which is usually, for example, 150% for vitamin A or 160%. But what we found that only couple product, chewable gel product can release, using dissolution procedure, develop dissolution procedure, can release almost assay value. But in majority case, they barely can meet 75% from label claim, which is shows they can release only 50% from assay value. So there is some, we found that it is dissolution test is extremely important for chewable gels, especially without clinical study. You know, we would like to be sure, at least USP, because our monograph is just voluntarily, because we have plenty products on the market who just don't use our monographs. So we just would like to be sure that at least the manufacturers who would like to meet USP requirements, I mean, it's can be relatively, I mean, I would consider high quality, but quite have decent quality in order to be sure that their product not only tasty, but also can release and provide some benefits for consumer in agreement with the label claim.
Thank you. And we have a very interesting question for Dr. Lee from the audience online. So, are there any special packaging considerations for 3D printed drugs? And also, it was mentioned that the tablets get individual QR codes. How would a consumer track which tablet was taken if there was an adverse event, since consumers don't always take the product in a systematic way and could report one tablet as a cause when it may not be the case? There is another part to this question, but maybe you can take the first part.
There are multiple parts. Let me just take the last part I remember
okay.
to start with. It's about the QR code and how would patient track it. And of course, from back end, we harvest a lot of data, including the manufacturing data and QC data. By the way, this production line I show you, there are 7 PAT points. So those are all taking the data at real time. But from the patient side, it really depends on what information they needed, probably some of the QC data or manufacturing data. But I'm not so sure what the purpose of the question is. If there's an adverse effect, you
no longer have the QR code because you consumed the product, right?
It's on the packaging.
It's on the package.
Yeah,
okay.
So you can go back to track that one if you don't throw it into the garbage can.
Okay. And are there any special packaging considerations?
Currently, we're using the aluminum blister packaging.
Okay.
Yeah. I believe Aprecia is using the blister packaging too. Yeah.
Okay.
They have a new technology, have a preformed cup, and you can have a powder in there while the printing. Yeah, that's a very neat technology too.
And how could the OTC industry utilize this more individualized tracking to enhance the adverse event reporting? I think you already touched upon this question. Yeah.
So just
from the
same thing. Yeah.
From the packaging.
Right.
Yeah.
There are probably some concern about the cost of manufacturing, which I probably will add into it. The current manufacturing cost is at a cent. If you take the API out of picture, the excipient and the manufacturing cost is at cents to tens of cents. So it's not like a lot of people think, creating a 3D printing product is going to be extremely expensive. It's not, yeah.
Okay. Can we take one more question, or we're out of time?
Could I add?
Sure.
Because it was question about mastication. So, I know that some manufacturers, when cannot meet the dissolution requirements, they said, okay, it's supposed to be chewable, so we just can cut them and then put to the dissolution vessel, and then it's measured dissolution. So actually, in this case, we follow FDA guidance for chewable tablets. So there is no any recommendation for cutting or in order to, because we cannot. It's the number of biting. It's not standardized, So and it's not possible to standardize any dissolution procedure, but using cutting of chewable gel. So we recommend to dissolution of whole piece. Without any cutting, which stimulates the sort of chewing. So thank you.
Thank you. I think we are at time. Back to Brandon.
Thanks again, Denai, for moderating. So that will take us. Oh, also wanted to say thank you to all of our panelists for that informative discussion. And that takes us to our break, our first break of the day, which will go until 10:40 a.m. So again, just a reminder, there's coffee, tea, and snacks available at the kiosk right outside of this room in the main hall.